Welcome. We are learning about the factors affecting expression of the clone genes in the E. coli. Today we are going to learn about the translational initiation sequences, how it is going to affect the expression of the clone genes in the E. coli. Translational initiation sequences, it is nothing but it is about the 5' UTR, that is the 5' untranslated region. In the previous video, we have learned about the promoter strand transcriptional termination. The link for this, it is provided into the description box. The upcoming videos, it is going to cover about the plasmid copy number, plasmid stability, host cell physiology, codon choice, and the mRNA structure. So going towards the today's topic, which is the translational initiation sequences. At the 5' UTR of the mRNA of the prokaryote, that is the bacteria, it is consisting of the Shine-Dalgarno sequence, that is the ribosome binding site. It is affecting the expression of the clone gene. Second one, it is a spacing between the SD sequence and the initiation codon. And the last one, it is a basis following the SD sequence. These are the factors which are going to affect the translational efficiency of the cloned genes. Now going towards the first one, it is a ribosome binding site or the Shine-Dalgarno sequence. Okay, so as the name suggests, the ribosome binding site means what? It is a site where the mRNA, it is going to interact with the ribosome. In the ribosome, there is a presence of the 16S rRNA, which has the complementary base sequences for the mRNA. Okay, for the mRNA, and on the mRNA, the bases, okay, the conscious sequences which are present, it is the AGGAGG. Here, AGGAGG, and this sequence, it is complementary to the 16S rRNA. Now, when there has to be the translation, the mRNA, it should interact with the ribosome, because the ribosome, it is the site where there will be the translation, that is, there is the protein synthesis, it is the site for the protein synthesis. So it is important that the mRNA, it should interact with the ribosome, and here it is interacting with whom? It is interacting with the 16S rRNA. Why these particular base sequences they are required? Because here when you look at the ribosome, the 30S subunit of the ribosome, in that there is a presence of what? There is a presence of this P site and there is a presence of the A site. P site, it is the site where the first, where there is a first incoming of the charged tRNA, it is a FMET charged tRNA. Okay, so here when there is an interaction between the SD sequence and the mRNA, at that time the AUG codon, which is a start codon, AUG codon, which is a start codon, it has to be placed properly over the P site. For this purpose, it is important that the SD sequence, it should interact with the ribosome, and when that interaction, it is efficient one, at that time only there will be the proper placement of the AUG over the P. So as the initiation of the translation, it is an efficient one, the translation efficiency, it is also going to be increased. If translation initiation, it is going to be a weak, the interaction, it is going to be a weak, at that time what will happen, that AUG, it is not going to be properly placed over the P, and the efficiency of the translation, it is going to be a lower one. The another factor which is going to affect the translational efficiency, it is the spacing between the SD sequence and the initiation codon. Now here it is the SD sequence and here it is the initiation codon, it is a AUG, and the SD sequence and AUG, it has some intermediate base sequences, and that it is called as the downstream box or the downstream region. And this downstream box or the downstream region, it is consisting minimum 5 to 10 bases. Minimum 5, maximum 10 bases it should be present. If it is lower than the 5, then the efficiency of the translation, it is going to be a lower one, and if it is going to be more than 10, then also it is going to affect the translational efficiency. And by observation, okay, it is observed that the 8 bases, it is a optimum one. Okay, that is when SD sequence it is separated from the AUG by the 8 bases, at that time efficiency of the translation it is a maximum one. Means what? This, okay, this AUG it is efficiently placed over the P. Okay, this AUG it is efficiently placed over this P side, so that the translation efficiency it is going to be a higher one. Next one, it is the basis following the SD sequences. Okay, so here this it is the SD sequence. Okay, UUGACCU. These are the bases. In this basis, if there are the 4 residues of the A or the 4 residues of the T, if they are present, this is the SD sequence. After it, okay, instead of this 4, okay, instead of this 4, if there is a presence of the 4A or the 4T, then at that time the translation efficiency it is the highest one. And if, okay, and if there is, okay, and if there is a placement of the 4C residues or the 4G residues, at that time the translation efficiency it is going to be increased by the 50% or the 25%. Okay, so in this way, okay, in this way, the 5' untranslated region, it is going to affect the expression of the gene. Okay, it is going to affect the expression of the clone gene in the E. coli. The first one, it is there should be the proper the conscious sequences of the SD sequence. There has to be the minimum of the 5 or the maximum of the 10 and the optimal 8 bases which are separating the SD sequence from the AUG codon. And the next one, it is a bases which are following the SD sequence. They are the important one. Okay, so I think you may have cleared the idea about the translational initiation sequences. If you like my video, please subscribe my channel and also share my videos with your friends so the knowledge it is going to spread. If you have any doubt, if you have any comment, then write down into the comment box. Okay, thank you.